Okay, we're now going to talk about the Golgi apparatus. Now the Golgi looks like a pile of flattened membrane-enclosed sacs. These sacs are called cisternae. The Golgi can be broken down into two different segments. There's the cis-Golgi network. This is going to be closest to the ER. And then there's the trans-Golgi network, which is going to be closer to the cell membrane. So when proteins enter, or when vesicles enter into the Golgi apparatus, they'll do so as they are put into vesicles from the ER. And then they'll enter into the cis-Golgi network. They'll be modified in the cis-Golgi network. They can be put through to the trans-Golgi network, and then they'll be further sent on to their destination organelle. Now, both the cis and the trans network are considered important for protein sorting. So if a protein enters into the cis-Golgi network and it has an ER retention sequence, the protein will be moved back to the ER. And if a protein leaves the trans network or the trans-Golgi network, it will be sorted to whichever organelle it's destined for. Now, remember that when we last talked about modifications that can happen to proteins, we talked about how oligosaccharides can be added in the ER. Now, these oligosaccharides that are added to proteins to make the glycoproteins can be further modified in the Golgi. And you can tell if a modification is going to happen early. So if it happens early, it'll happen in the cis-Golgi network. Or if it happens late, it'll happen in the trans-Golgi network. And the way that you know whether a modification will happen early or late, or in the cis or the trans network, is where the location of that modifying enzyme is. So if the modifying enzyme is in the cis network, and it further modifies that oligosaccharide on your protein, you could say that it's an early modification. So we talked a little bit about the constitutive exocytosis pathway. So we're going to leave the Golgi apparatus. And leaving the Golgi apparatus in vesicles is a steady stream of vesicles that bud and fuse with the plasma membrane. Now, this is known as the constitutive exocytosis pathway. Now, constitutive means that it's just happens, it's not necessarily regulated. It's also known as a default pathway, as proteins that lack any signal sequence after leaving the ER. So that means that they don't have a signal sequence that would send them to the lysosome, or they don't have an ER retention sequence that would send them back to the ER. So if they lack a signal sequence after leaving the ER, then they'll be put into this pathway. And that's why it's called the default pathway as well. Now, if we look at this figure, what the constitutive pathway is shown in pink, I think. So another part of constitutive exocytosis is secretion. And the secretion is the process of releasing products, which can be proteins or other components, to the outside of the cell. So this vesicle is going to bud off of the Golgi, and it's going to move to the outside of the cell. And when this vesicle fuses with the membrane of the cell, this is what's going to empty the contents into the extracellular space. Now, not everything in the cell is going to exit the cell in a constitutive way or in a steady way. Another pathway that can be used is called regulated exocytosis. Now here, proteins are going to be diverted into secretory vesicles until an external signal is received. And so a good example for these are hormones or neurotransmitters. So neurotransmitters are held in vesicles at the terminus of a neuron until it receives a certain signal. And when it receives that signal, that's when all the vesicles are going to fuse with the membrane, and that's what's going to empty the neurotransmitters into the synapse, which is the area between the two cells. That's regulated exocytosis. So some cells are specialized cells that will make large large quantities of particular products, and these cells are called secretory cells. So the example I gave you before was a neuron that makes a lot of hormone. And then that hormone is going to be stored in a secretory vesicle for later release. So again, a secretory vesicle is simply a storage vesicle that holds large amounts of cargo for later release. Now the cells that are going to use secretory vesicles, they'll produce a large quantity of product, and that could be a hormone if it's a neuron, it could be a mucus, it could be mucus if we're talking about goblet cells in the immune system, or it could be digestive enzymes that could be held in these secretory vesicles. Now, these secretory vesicles are going to bud off from the trans-Golgi, and they're going to accumulate near the plasma membrane, but they're going to be held in stasis there, or held still, until an extracellular signal stimulates them to fuse with the plasma membrane and release their contents into the cell exterior. So if we look at the picture in this electron micrograph, what we're seeing is a secretory vesicle, and in this particular secretory vesicle, where it contains insulin, here's the plasma membrane, and what we can see is that this cell has received a signal to have these secretory vesicles fuse with the plasma membrane, and this is what's going to cause the contents of this secretory vesicle to be emptied into the extracellular space. So this is a vesicle that has fused with the plasma membrane. So this insulin is going to be secreted or released. So I just want to go over a few techniques that scientists use to track and sort proteins. And they use these techniques not only to track proteins, but also to figure out how proteins are sorted and where these proteins are going to go within a cell. So we already saw an experiment where you swap the signal sequence on one protein and put it on another protein. And in that example, we had the ER signal sequence, and we said that if you put it on a protein that's normally not bound for the ER, then that's enough to put that protein into the ER. And conversely, if you remove that signal sequence from a protein that should be bound for the ER, that protein will remain in the cytosol. So a way that scientists first started looking at these signal sequences, or where the proteins are going to be, was done a few different ways. So there's a biochemical, a genetic, and a molecular biology way to track these proteins. We're going to go through the biochemical way first. So what you're going to do with this is you're going to have a protein that either has a signal sequence or it doesn't. And that's what's shown up here. The signal sequence is in red. And what you're going to do is you're going to radiolabel your protein. So that's how you're going to be able to track it. So you have your protein that's radiolabeled, and then you're going to incubate it with the organelle that you isolated. Maybe you isolated a mitochondrion, or maybe you isolated the ER. So after this incubation process, if your protein has the appropriate signal sequence, it should move into that organelle. If your protein does not have the appropriate signal sequence, it should remain outside of that organelle. So the next step is to try to figure out if the protein entered into the organelle or not. There's two ways to do this. One way is you could just take this mixture and centrifuge it. And if you centrifuge it, what you expect to find is that the heavier portion, which is going to be the organelle, is going to be at the bottom of the tube, and the lighter portion, which would just have free protein in it, protein that did not enter your organelle, would be at the top. And then if you check to see where your radiolabel is in this tube, that would tell you if your protein is a free protein, and in that case it did not have the correct signal sequence to enter into that organelle. Or if it's at a heavier portion on your tube, then that would tell you that your protein did enter into the organelle, because it would be associated with the organelle in this portion of the tube. The other way that you could test this is by using SDS gel electrophoresis. So again, we're going to go back to our mixture, which either has our protein inside the organelle or has our free protein if it doesn't have the correct signal sequence. So if you take this mixture and you add protease to it, what should happen is that any protein that is outside of the organelle will be degraded, and that's going to be degraded by the protease. Now, if you have your protein inside the organelle, it should be protected. The way you can verify that your protein is indeed inside the organelle is now you can take that organelle with your protein inside of it and you can add detergent to it. When you add detergent to it, you're going to disrupt the organelle, you're going to disrupt the membrane of the organelle. And if you also add more protease at this step, now you should degrade your protein. Now, what I want you to do, and what would be nice if you would turn in on your paper that you have your 3 questions on, is draw what you expect this gel to look like. So again, it should be a SDS page electrophoresis, and what you want to do is your first lane should be your protein with your organelle. Your second lane should be your protein with your organelle plus protease. And then your third lane should be your protein plus your organelle plus protease plus detergent. And you should draw both scenarios where you expect your protein to be inside the organelle, draw the bands that you would expect to see, and then draw the scenario where your protein is not inside the organelle, and tell me what bands you see. And the hint I'll give you is that when proteins are degraded by a protease, they make several small bands near the bottom of the gel. If you have intact protein, it will make one large band near the top of the gel. We'll go through this in class, but it'd be nice if you had pictures already drawn out to do this too. Okay, so that was the biochemical approach, and it's biochemical because we had isolated components of the cell. The next approach that we're going to talk about is genetic analysis. So in this type of approach, what we're going to do is we're going to search for proteins that are important for different steps of exocytosis. And if we mutate these proteins at different steps, it's going to have different consequences for the cell. So what we're going to do is we're going to make a mutation, and then we're going to see where things start to accumulate in the cell. And by doing this technique, you can identify the majority of the genes that are involved in exocytosis. So we're going to look at the normal cell first. So here we have a protein that's destined for secretion. It's the light blue dots. This protein is inserted into the ER. Then it's going to be bud off in a vesicle, move to the cis Golgi, then through the trans Golgi, and from there it's going to enter into a secretory vesicle, and then it's going to undergo exocytosis. Now let's say we have a mutant, and this mutant, we'll call it secretory mutant A, where when we have this mutant, it's going to cause all of our protein to accumulate in the ER. And what we can glean from this is that the protein secretory mutant A is important for vesicle formation and transport from the ER to the Golgi. Now another mutant that we may have, we'll call it secretory mutant B, could cause an accumulation of protein in the Golgi. And if this was the case, we would make the assumption that our secretory mutant B is important for vesicle budding, leaving from the Golgi to the plasma membrane. Now, the way that you do this is you use temperature-sensitive mutants. And with these, this is commonly used in yeast. What happens is that when you keep the yeast at 25 degrees C, all the genes are going to be completely functional. But when you shift them to a higher temperature, like 35 degrees C, that's when the gene, which is temperature sensitive, is going to become inactivated. So that's one way that you can do this genetic analysis. And then what you do once you shift the temperature is you look for where the protein is going to accumulate. So here we had it accumulate in the ER, and here it was accumulated in the Golgi. Third technique is molecular biology with microscopic analysis. And here, what we're going to do is we're going to tag our protein with GFP. GFP stands for green fluorescent protein. And what we can do then is we can look at our cells under a fluorescent microscope. And if we use a fluorescent microscope, we'll be able to see where the GFP is. And again, that GFP is going to be linked to our protein. So in this experiment, we can just see various effects of having our GFP. So what we're looking at is a viral coat protein. So in an infected cell, proteins tend to move from the ER to the cell surface. And that's where at that point the virus would be assembled. Now in this experiment, the protein that we're interested in has a mutation that only allows it to be moved from the ER if the cells are at a low temperature. At high temperatures, it will remain in the ER. So figure A is a high temperature, and this is going to show us where the ER is. So here's our nucleus, here's our ER. The protein's stuck in the ER because this is a high temperature, and we made a temperature-sensitive mutation. Now, if we take this cell that's at a high temperature and we lower the temperature, which is happening in figure B, the protein starts to accumulate in the ER exit sites. So you see this punctate staining. Punctate is a fancy word meaning that you have these bright little spots of GFP in the ER now. That shows you that the protein is being put into vesicles and it's going to be moved out of the ER. Now in C, we're following just a little bit further along. C, we have the protein moving to the Golgi. In D, the protein's going to finally be delivered to the plasma membrane. And the arrowheads are showing you right here, here, and here, where particularly not the pink arrows, sorry, the white arrows are showing us where one vesicle has successfully fused with the plasma membrane. Okay. So that was exocytosis. And we're going to talk briefly about endocytosis now. Now, with endocytosis, this is how cells are going to absorb molecules by engulfing them. And we can break endocytosis down into two different components. There's penocytosis, peno meaning very small, which literally translates into cellular drinking. And this is the ingestion of food and molecules that are less than 150 nanometers in diameter. So cells continuously undergo penocytosis. This is how they sample their extracellular environment. Now the other type of endocytosis is referred to as phagocytosis, and this translates into cellular eating. And in cellular eating, cells ingest a large particle, a particle greater than 250 nanometers in diameter. Sorry, I'm battling with my cat right now. I was trying to sit on my computer. Okay, so phagocytosis. Now, only certain cells undergo phagocytosis, and these cells are designed to do this. And so one type of cell that will undergo phagocytosis is macrophage. A macrophage is a professional phagocyte, and a phagocyte is just a cell that specializes in phagocytosis. So that's what we're seeing here. So here's our macrophage, and what it does is it extends its cytoplasm to surround a bacterium or for an invading cell. And so here's two bacterium that this macrophage is surrounding, and what will happen is that these extensions of the cytoplasm will fuse on either side of this bacterium, and then the bacterium is going to be put inside of that macrophage. Eventually, it's going to fuse with the lysosome, and that's how the bacteria is going to be degraded. Now, it doesn't have to be just foreign cells that are going to be phagocytosed. Our cells in our body will also phagocytose other cells that are damaged or worn out. So here we see another phagocyte. This phagocyte is digesting red blood cells. So these red blood cells are worn out. They need to be destroyed and recycled. Now, penocytosis is really an indiscriminate process. So whatever is nearby can be trapped and brought into the cell. And again, this is how the cell is just sampling its extracellular environment. But it can also be a specific process. And when it is specific, we refer to it as receptor-mediated endocytosis. Receptor-mediated endocytosis uses clathrin-coated vesicles. Now, specific molecules can be picked up this way when they bind to their receptor on the cell surface and enter the cell as a receptor macromolecule complex in a clathrin-coated vesicle. This helps the cell take up specific molecules and concentrate them without drinking large volumes of extracellular material. Now, an example of this is in cholesterol uptake. So, cholesterol is insoluble, and to transport it into the cell, it has to be transported by a protein. And the protein that does this, that helps transport cholesterol, is called low density LDL, or low density lipoproteins. And when we have cholesterol with it, we call it LDLC, for low density lipoprotein with cholesterol. Now, in this figure, we see the LDL, which is the low density lipoprotein, binding to its receptor. Now, this receptor is going to be taken up into the cell by receptor-mediated endocytosis. It's going to be put into a clathrin-coated vesicle. The clathrin is going to uncoat, so we'll have a naked vesicle. And then this vesicle is going to fuse with an endosome. Now, I told you that the endosome is the recycling center of the cell. What the endosome is going to do is it's going to separate the LDL from the receptor, and the receptor is going to be recycled back to the cell surface through exocytosis. Now, the LDL, which again has the cholesterol molecule with it, is going to fuse with the lysosome, and here the LDL and the cholesterol are going to be released from each other. And the cholesterol is going to be removed from the lysosome, and then the cell has free cholesterol now that it can work with. Okay, so in receptor-mediated endocytosis, especially with the LDL cholesterol, we saw that when it entered into the cell, it went to the endosome. Well, endocytosed macromolecules are sorted into the endosome, and usually they'll enter first into what we call the early endosome. The early endosome is going to be found below the plasma membrane. So that's what's right here. Now, later on, other molecules can move to the late endosome, and the late endosome is going to be found closer to the nucleus. Here's the nucleus here. I don't see a great picture of a late endosome on this, but you can imagine another endosome sitting over here. So, if we're talking about an endocytosed receptor, there's three possible fates. The first one we already talked about. So, when the LDL bound to its receptor and then entered in through receptor-mediated endocytosis, fused with the early endosome, we saw that the LDL was separated from the receptor, and the receptor recycled back to the plasma membrane. So that's one fate of endocytosed material. It can just be returned to the plasma membrane. Another fate, which is what we saw for the LDL receptor complex, or LDL cholesterol complex, is it could be targeted to the lysosome, and if it's in the lysosome, it's going to be degraded. A third fate is called transcytosis, and this is where the vesicle is going to travel with its cargo to a different part of the cell. So here it's showing you and fusing with the basolateral membrane instead of the apical plasma membrane. So we talked about lysosomes when we began chapter 15. We want to talk about them again now. So the lysosomes carry out controlled intracellular digestion of both extracellular materials as well as worn-out organelles. And we said they contain lots of enzymes, so nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases, phospholipases, all of those are listed here. And we can find them all within the contents of a lysosome. Now, I also told you that the lysosome lumen is acidic, so it has a pH of about 5.0. It's acidic, especially when we compare it to the pH of the cytosol, which is 7.2. Now, there's pumps in the membrane of the lysosome that actively keep it acidic by pumping hydrogen ions inside. And these enzymes, again, are only going to function at this low pH. Now, I also told you that this low pH is also a safety mechanism, because if this lysosome were to rupture, these enzymes are only going to work at a low pH. So if this lysosome ruptured and it released its enzymes into the cytosol pH is too neutral for them to function. So it's a really good safety mechanism, so that one ruptured lysosome is not going to kill a cell. Now, there's also transporters that we find in the lysosomal membrane, and that's going to allow products to be moved from the lysosome to the cytosol. So we saw this happen for cholesterol. Okay, so cholesterol was an example of a material that came from from the extracellular fluid of the cell. Now, the lysosome is also going to degrade worn out organelles. And when the cell needs to destroy its worn out organelles, the process is called autophagy. And in autophagy, the organelle is going to be surrounded by a double membrane, and this is going to form a structure that's called an autophagosome. And then what's going to happen is it's going to fuse with the lysosome, and this is how the organelle is going to be degraded. So again, this process is called autophagy, and it's specific to the destruction of cells' obsolete organelles. And in this example, we're using a mitochondrion. Okay, this is the end of chapter 15, and I will move on to chapter 16 now.